whether it's for a long-term storage, a short-term storage, or whether you need to do your analysis on freshly prepared sample. For analysis, working with EVs has a lot of options and also possibilities and all of that. But first of all, you need to consider, should I isolate my vesicles prior to the analysis? And how should I isolate it? Should I do it by size? So I'm using size exclusion. It could also be a combination with the density gradient and also centrifugation. You could also consider charge. It could be whether you need an anion exchange solution for that, or whether you need to go for specific surface markers. It could be using antibodies or certain lectins. It could be by beads, but also unit precipitation. And then again, also just pointing back is that should you do your isolation procedure before you store your samples, or the opposite way around? That is also really need to concern within this area. So we have the type of analysis. That is, of course, depending on the molecule that you would like to analyze. Working with EVs, you can either choose to go with the surface proteins. It could be the transmembrane adhesion molecules or particular receptors within a biomarker. You can also work with the soluble proteins, the enzyme or the signal molecules. You can also, another part is looking at the lipids, the phospholipids, the cholesterol, etc. But you also work into the nucleic acid parts, the DNA, the microRNA, and the messenger RNAs. Or you can choose to work within the size and the numbers, for instance, using the nanotracking analysis. Doing your data analysis afterwards, of course, depends on which type of analysis that you have available in your lab. But you can choose either to do kind of a targeted analysis or do a more discovery-driven analysis. Within the targeted analysis, we have particularly the immune acids, as well we know. We have all done our Western blots to see whether we can find a particular marker, but it could also be an ELISA or an EV array. Another option is also to do a more targeted mass spec, so the SRM or MRM. It could be either label-based or label-free. And also, if you're looking for the nucleic acid, it could be the RT-PCR or qPCR. In the discovery part, you have, of course, the proteomics part. You can do a top-down analysis or bottom-up analysis. For the nucleic acids, you can do the next-generation sequencing, depending also whether you want to do kind of whole genome or exome sequencing, but you can also choose to do a microarray screening part. Then coming to the next part is to do your analysis, which is what kind of analysis do you need to find your biomarker. You can go for a simple, up here we have a simple linear model, or you can do a more comprehensive study. You can do machine learning, or even further on into deep learning, deep neural network. You can do an AI kind of analysis. But what you need to consider is also the opposite road over here, so that if your interpretation of your analysis is easier if you do a simple model than if you do a more complex model. It can be really hard to figure out the biology behind your results if you need to do a deep learning neural network to be able to find your biomarker. Within the biomarker discovery, again, you need to see how you can best separate your groups so that you have the best true negative part or the best true positive part. And then, of course, you will also find some having false negative and false positive results. Generating these receiver operating curves so that if you have an AUC of approximately 95%, then you have a really excellent biomarker, or just what we normally call a good biomarker. And this is actually where most studies actually stop and people start to publish their results, because now they have actually found the biomarker. But one of the most important steps is actually the following validation of your results. And the validation part is you can actually split it up, starting with an analytical validation. So in some way you need to develop a quantitative assay. You also need to validate this kind of quantitative assay and then assess the range, specificity and sensitivity of your assays before you then can go into a kind of clinical validation where you need an independent patient cohort. You need to assess specificity and sensitivity of the entire analysis, but also you need to assess the utility, the robustness, and also viability. And then you can end up developing a kind of clinical assay on that. So following this part through an entire biomarker study is a long way, first of all, but actually there are tools for you to assist within, and also because when you start a biomarker study, we all want to kind of see if we want to save the world or at least the patients. And to do that, we also need to bring it further on into the clinic part. Within EU, we have had a project where TTOs have actually developed this free tool. It's available on biomarker.nu. And it's actually an online tool where you can put in all your data in your discovery phase, also in your verification phase. And this one, Mark Green, is within the research part, but it takes you to see when is it that you need to consider to generate a prototype of your assay and how to validate their performance. Then it can take you further on because this is kind of related to the technology level of readiness. When should you transfer it, and also how can it come to the market? And particularly, they have this kind of paragraph kind of set where you also go into all the regulatory considerations when you're doing the biomarker study. So it's a free tool to use. So there are a lot of published studies already, but I've looked further into what's actually registered as clinical trials at the moment. 159 studies are involving using extracellular vesicles for detecting and measuring various diseases. And as you can see here also, and I think this curve is quite similar to the number of publications as well, but also the number of registered studies is also increasing. And it should be said that actually most studies found in this 159 studies is only 40% of the total part. Most studies is actually using EVs as therapeutics. These biomarker studies, they have of course different sizes of cohorts, number of enrolled patients. Most are within these groups within 10 to 100 or 500 patients, and actually a few of the studies is also including more than 1,000 patients. And if you can see here, we have the number of studies per country. So US is in the top, but as coming from a small country as Denmark, I'm actually proud that we are in the top 5 list here of countries with registered clinical trials. And looking into the distribution of these studies, cancer is the most dominant one. Then we have within metabolic diseases, and a lot of these metabolic diseases, I would say 1/3 or so, is actually within obesity. Also a lot of neurological studies is going on, and then we have some smaller numbers within other disease parts. So if we look further into this group with cancer, I can just recommend this review where you have a lot of cancer biomarkers put in. You have a lot of different cancers, and it's also both you have nucleic acid reports, but also within proteins and all of these different cancers. Going further on to some of the other disease groups, we have this review of Das and co-workers, where you have split it up both into the neurodegenerative diseases, infectious diseases, diabetic and cardiovascular diseases, and kidney diseases, of course, besides the cancerous. So the session today is actually also, some of them are within the cancer part. We have 4 talks with cancer, We have one with breast cancer using nanoflow cytometry, and then we have some short talks. First with the digital droplet for diagnosing cancers, and then we have Serene talking about saliva exosomes for oral cancer detection, and also again we have some with the ovarian carcinoma. Another group of presentations that we have today is within reproduction and metabolism. We have gestational diabetes being presented here, and also endometriosis presented. So summarizing all of this, we can say that we have some future challenges. So we need to ask ourselves, what is the current unmet needs before we start our biomarker studies? But we also need to face that we have kind of lack in standardization. It's still a problem. And within this review article, Gonzalez and co-workers is actually going through the challenges and opportunities for clinical laboratory working with EVs. And then we also need to consider that we have a lot of patient heterogeneity. We are different people, we are different populations, and we need to face that as well. So, in summary, this is short, is that we need to figure out who we need to find in our study. Then we need to see where do we search for our biomarker, where should we look for it? Is it in the bloodstream, in the urine, or for Wally here on the beach side? Then we need to find the right tool to use to find our biomarker and see how to search for our biomarker. Which tool is the most appropriate one to use for this particular biomarker? And then we can actually start searching for our biomarker here. Ending up finding out, okay, we found our biomarker. Next step is actually, how can we catch our biomarker? How can we make this into a clinical development part and get this biomarker out? Which tools or transport should we actually use for this? Or should we go with an old-fashioned kind of rocket methods? But most important is that we need to catch our biomarker and find the right biomarker for our disease. When we've done that, we can start covering the campaign. We can then start writing our publications. And then I just want to say that hope that you enjoy the rest of this session, and thank you very much.
Thanks, Melanie. My name is Soumya. I'm from the University of Queensland. I'm chairing this session with Melanie. So we would like to introduce our first speaker for this session, Yun Hu from Xiamen University. She'll be talking about enhancing breast cancer diagnostics and subtyping, multiplexed profiling of dual surface protein expressing single extracellular vesicles via nanoflow cytometry. Welcome, Yun.
Thanks for the introduction, Stanley. Hello, everyone. Good morning, everybody. My name is Yun. I'm from Xiamen University, and I'm very happy to be here to share and talk with you. And the topic is enhancing breast cancer diagnosis, prognosis, and subtyping, dual surface protein profiling on single evase and nanoflow cytometry. There are 4 parts involved. Breast cancer stands as a critical global health concern worldwide, and is highly heterogeneous and dynamically variable. However, traditional screening techniques such as mammography or tissue biopsy were light-sensitive, highly invasive, and time-consuming, which severely hinders the early detection and precisely treatment implementation for breast cancer patients. Thus, developing rapid and effective methods for early diagnosis, prognosis, and subtyping BCA are of paramount importance. As we all know, extracellular vesicles has been recognized as one of the hottest liquid biopsy candidates recently. And actually, there are multiple evade proteins have been recognized as diagnosis and prognosis biomarkers for breast cancer. However, in these literatures, the either example averaging methods were employed or only one protein marker were analyzed, both failing to identify EV subpopulations directly from tumors with enough sensitivity and specificity. Well, it has been reported that multi-protein analysis on single EVs could represent a solution to improve the clinical diagnostic efficacy because it can classify EVs into different subpopulations and to improve the specificity in origin and function. And in our lab, we have also developed an advanced platform called nanoflow cytometry, enabling multi-parameter analysis of single EVs as small as 40 nanometers. And through the particle concentrations of EV subsets expressing specific proteins, we have achieved the early diagnosis and prognosis of colorectal cancer, as well as the differential diagnosis of nasopharyngeal carcinoma and nasopharyngitis. So based on all the background, we plan to perform dual surface protein profiling on single EVs via NFCM and to classify EVs into different subtypes, aiming to improve the clinical efficacy of early diagnosis, prognosis, and subtyping. So firstly, we examined the EVs from breast cell culture in vitro. And through extensive literature researching and another single particle techniques called proximity barcoding assay, we have chosen 6 proteins for downstream validation. But in consideration of that PBA technology, principally simultaneously detect 200 proteins at a time. So there might be potential steroid hindrance by multiple antibody staining. Thus, we exploited the NFCM to validate the expression levels for every single protein in single particle level at a time. Take IgG1 for example. NFCM could simultaneously reveal the expression proportions and the median fluorescence intensity. Overall, we found that all these candidate proteins can indeed serve as potential markers for at least one subtype of breast cancer. However, for single protein markers, no matter through the proportions or MI, neither can we differentiate EVs from 3 different BCA subtypes, nor can we distinguish between BCA EVs and EVs from other tumor regions or normal cells, such as the red blood cell. Thus, we have to involve more parameters and then return to dual protein analysis. And PBA technology has also helped us choose the IgG1 and the CD151 as the most highly expressed protein combination across all BCA EVs. And because it became and HER2 are 2 widely reported protein markers for breast cancer, so we and it also showed, the combination also showed significantly overexpression on MCF7 and SKPR3 derived EVs. Similarly, we used exploited an FCM with a strategy with light steroid hindrance to validate their expressions. We used 2 4 dye-conjugated antibodies for recognition and used an FDM equipped with 2 lasers for analysis. And for the from the 2 fluorescent signals, we could classify the detected EVs into 4 distinct subtypes and each has 3 FL parameters, including the percentage and the median fluorescence intensity for IgG1 or CD151. Let's take IgG1 and CD151 example. Yes, yeah. But for EVs in Corrigan 2 and in Corrigan 4, only one fluorescence median signal is comparable. Thus, we just extracted 7 fluorescence parameters from each EV sample and conducted the Tisserning analysis, and we observed that distinct differentiation of the 4 EVs, including 3 breast cancer subtypes and the normal cells, can be differentiated. But we can also characterize the 2 proteins in 2 separate samples, and we also collect 4 fluorescence parameters, but no matter how we optimize the perplexity of the Tisserning analysis, we could not distinguish the normal EVs from CF7 direct EVs. So this underscores the clinical potential of dual protein analysis on single EVs for BCA subtyping. And for clinical samples, we firstly characterize the particle concentrations of 6 EV subtypes in plasma from 9 healthy donors and 20 breast cancer patients with early stage. And we found that the unweighted sum of the concentrations of 6 EV subtypes outperformed every single EV subtype and total EV numbers for early diagnosis, and AOC achieved 1. That means protein phenotyping and the conjoint analysis of several EV subtypes could improve the diagnosis ability. But in that case, for each patient, I have to conduct 6 parallel samples. That is so much time consuming. So we just we want to characterize 2 proteins simultaneously in one sample. And at a single particle level, let's take EPKIM and HER2, this combination, for example. After subtyping, we could get more information because there are EPKIM positive, the concentrations of EPKIM positive EVs, or HER2 positive EVs, or the unweighted sum, and the concentrations of each subpopulation in PEP2 or P4. And we found that it was the EV subpopulations rather than the unweighted sum always have the optimal discrimination for all protein combinations we tested. And specifically for this combination, we found it was EV only expressing HER2 rather than the EV subtypes co-expressing HER2, and EPKIM had increased the diagnosis capability. And so then we also explored the prognosis capability for breast cancer, and we found similar results that it was IgG1 positive, but CD151 negative, or EPKIM positive, HER2 negative subpopulations exhibited a stronger prognostic capability. So there comes to the conclusions. Well, through the dual surface protein profiling at the single EV level in XAM, we firstly utilizing breast cell line EVs as the model and utilizing 7 fluorescence parameters to conduct the Tisserning analysis. And we found that we could achieve the effective differentiation among EV from 3 BCA cell lines and normal breast cells. And for clinical samples, we found the particle concentrations of specific EV subpopulations always have improved efficacy for early diagnosis and prognosis. And we had to suggest that it might be the EV cluster with only one marker rather than double positive EVs might be more important. And in the future, we will further increase the numbers of clinical samples to validate our results just now and to investigate the classification ability of differentiate the different BCA subtypes. And we hope our methods will advance the disease monitoring and the precise treatment for BCA patients. Okay, that's all. Thanks for your listening. Welcome to Xiaoben. Thanks.
Thank you, Yunren. Do you have any questions?
Thanks. I'm just trying to get my head around all the fascinating data that you have presented. But I have one question with regards to, you demonstrated that EpCAM positive and HER2 positive gave a poor prognosis, and you could use that to early detect with the AUC above 90. So my question is that there are triple negative patients, which means ER negative, HER2 negative. How do you detect those patients using the markers that you have demonstrated?
Yeah, thanks for your question. Yeah, it's a really great question. Actually, because we now just collected 20 breast cancer patients, and in consideration of that triple negative breast cancer and HER2 positive cancer just accounts of smaller proportions of all breast cancer patients. So in my clinical cohort, there are almost 60 or 50 nominal subtypes. So I just said that in the future I have to increase the numbers of clinical sample and to further investigate the classification ability. I don't know.
So no. The question is, can you use the markers that you have currently identified, which is the EpCAM and HER2, in early detection of a cohort that doesn't express these markers? We can talk later.
Okay. Thank you.
Thank you for the presentation. I think the technology is quite great. I just wondering have you considered because your samples are quite unbalanced, right? You have high control, you have 20 cases, so the incidence of the disease in your cohort is 66% or something like that, right? So that means it's quite unbalanced, and that means that potentially you're going to have affected your sensitivity or your method. So I just wonder if you're trying to balance the analysis using different, perhaps, classification models and how that may affect your sensitivity.
Let's try to explain your question. You mean how can I to improve the sensitivity in the future?
Yes. So I think the question is more a comment that you might consider to when you're doing your analysis. Your number of control is quite low, right?
Yes.
So that means that the incidence of the disease in your population is over 60%, which is not realistic, right? So my suggestion is you're trying to balance the data and have different techniques to do that, so you can increase the number of control that you have. Right?
Thanks for your suggestion. Yeah. Yes, EpCAM and HER2 have been reported.
So do we know their expression in non-EV components? Do we really have to do the EV isolation and look for these markers, or we can see this differential expression with the non-EV components? Have anyone reported, or do you have any idea of that?
Yes, and because we wanted to, and now this method could not differentiate the different subtypes. And just now I just talked with a clinical patient, and he said there is an urgent need to differentiate between different BCA subtypes, because different EV subtypes has different treatment. So we want to to know exactly what the breast cancer subtypes, and to implement precise treatment for her. Yeah, I'm not sure I answered your question.
Yeah, that's great. Thank you. you, Union. Let's put our hands together for Union. And I'd like to invite our next speaker, Nandini Jayabalan. She's from University of Queensland University of Technology, QUT, and she's talking about paracrine microRNA communication via extracellular vesicles, regulating glucose metabolism and fetal growth in gestational diabetes mellitus between maternal adipose tissue and placenta in vivo. Welcome, Nandini.
Good morning, everyone. Hope you all had coffee and ready for my short talk. So my study here is about investigating the role of adipose tissue-derived EVs in regulating placental function as well as fetal growth in gestational diabetes mellitus GDM pregnancy. So GDM is the glucose intolerance that is first diagnosed during pregnancy, affecting up to about 30% of all pregnancies worldwide. So although there's no one specific reason, but maternal obesity has been proposed as one of the major contributors. So what happens in GDM is the dysfunctional adipose tissue reported to secrete dysregulated amount of adipokines and have been reported to have an implication on the placental function. Now, the problem is not all obese women develop GDM, and secondly, there's always been inconsistency in data on the role of adipokines in regulating placental function. So this made us to think, what could be this one factor that's regulating placental function in GDM? And we hypothesized that it could be the EVs. So to test our hypothesis, we started off by characterizing the population of EVs in maternal circulation, and we found in GDM the majority number of EVs are coming from placenta and followed by from the adipose tissue. So we narrowed our focus to study the adipose tissue-derived EVs. So for that, we obtained omental adipose tissue from both normal and GDM pregnant women, performed the explant, isolated EVs from this conditioned media, characterized them, and we found the GDM adipose tissue releases higher number of vesicles, extracellular vesicles, and this number of vesicles positively and strongly correlated with fetal birth weight z-score. Not only just that, the GDM adipose tissue EVs upregulated the expression of genes associated with glycolysis and metabolism in the liver cells. So this made us to think, what could be this factor that is associated with EVs that is contributing for this? And we were particularly interested in studying the microRNA profile of these EVs. So for that, we isolated RNA from both the adipose tissue and EVs from normal and GDM pregnant women, performed small RNA sequencing, and did some comparative study. So it was really interesting that we found some distinct profile of microRNA that was only enriched in the EVs, and some of the microRNAs were highly expressed in the GDM adipose tissue EVs. So based on bioinformatic tools and literature search, we narrowed our microRNA candidates to 3 of them and performed qPCR validation study. And we found our 3 microRNA candidates were really highly expressed in the EVs compared to the adipose tissue or placenta, and even more highly expressed in the GDM EVs. So before we move on more with the microRNAs, we wanted to know if the adipose tissue EVs have any more role in regulating the placental function. So we did some in vitro study where we isolated primary trophoblast cells and treated the cells with EVs from both GDM and normal patients. And interestingly, we didn't find much of a difference between the normal and GDM adipose EVs in regulating the inflammatory profile of the placenta. But interestingly, the EVs from the adipose tissue, from GDM adipose tissue, upregulated the nutrient uptake, especially glucose in this placental cell. And we also confirmed this with proteomic analysis, where we found the GDM adipose tissue EVs treated cells showed upregulation of proteins that are associated with metabolism. So now we wanted to check, is this being regulated by our candidate microRNA? So we performed some transfection study where we transfected the primary trophoblast cells with our candidate microRNA, and again we found a similar kind of results where there not much of a difference in the inflammatory profile, but one of our candidate microRNA near 5155P upregulated the glucose uptake in these placental cells, and again the proteomic analysis showed the increase in the proteins that are associated with metabolism. Now that we have confirmed the adipose tissue-derived EEVs have an effect in regulating placental function in vitro, we wanted to confirm our data in vivo. So we injected pregnant mice with EEVs from normal and GDM adipose tissue, but we also loaded them with EEVs, injected them with EEVs that were loaded with our candidate microRNA. And excitingly, the EEVs from GDM patients as well as EEVs that were loaded with the microRNA caused and glucose intolerance in the pregnant mice, and we also increased the fetal weight as well as the fetal blood glucose level. So based on this, now we know the adipose tissue-derived EEVs have an effect in regulating placental function in GDM pregnancy, which can contribute to the nutrient uptake transfer to the fetal, causing fetal overgrowth or macrosomia, which is commonly observed in GDM pregnancies. And finally, I would like to thank the committee for giving me this opportunity, my supervisors, Carla Solomon and Martha. And if you have any questions or to know more about the data which is not here, please drop by my post. Thank you.
Thank you, Nandini. In the interest of time, we'll move to the next speaker. We have next a short talk from Huyen Lin. He's from Nanfan Hospital, Southern Medical University, and he will be, she'll be talking about profiling of single vesicle surface proteins via droplet digital immunoPCR for multi-subpopulation extracellular vesicles counting towards cancer diagnostics. Welcome.
Okay, good morning everyone. I'm Hui Xianlin from Southern Medical University in China. Today I'm so glad to be here to have 5 minutes chance to introduce our work. Profiling of the single vesicle surface proteins with droplet digital immunoPCR for the multi-subpopulation AV counting towards the cancer diagnosis. As we know, the EV is an important nanoscale circulating biomarkers for the cancer diagnosis. However, EV has enormous heterogeneity, especially in its origin and the biomarkers they carry. So the detection of the tumor EVs at low abundance is easy to be disturbed by the crowd of the EVs from the normal cells. So we think the specific EV subpopulation analysis technique is still urgent, and it is also what we exactly focus on. Through this background, we have developed an DDI-PCR assay for the multi-subpopulation EV counting, especially for those tumor EVs at low abundance, based on their protein profiling. In this assay, the EV will be labeled with the antibody DNA content firstly, and the protein message will be transformed to the DNA, and the DNA will trigger the triplex ddPCR for the EV subset analysis at the single EV level. In this way, the EV could be roughly divided into roughly 8 clusters based on the 3 kinds of the protein biomarkers. Here are some main results. Firstly, the activity of the antibody in the compound was verified, and we isolate the EVs through the ultracentrifugation and carry them by the TEM, NTA, and WB for further use. Then, we use the dual fluorescent detection channel with the well-designed probe concentration to construct the triplex ddPCR, and we conduct a systematic performance evaluation in this part. In the specificity validation, EV from different breast cancer cell lines were isolated, and they CD moleculars, HER2 and CAM were profiled in this DDI-PCR assay. And as the result shows, the EV from different resources can be distinguished well with high specificity. And we also evaluate its sensitivity. And the LOD of this assay could be as sensitive as less than 10 EVs per microliters. These results are consistent with the results of that of the NanoView, also verifying the accuracy of this assay. Then we collect the healthy and the patient blood samples for the evaluating its clinical application in cancer diagnosis. In this work, the breast cancer and the hepatoma carcinoma cohorts was used for the evaluation. As we can see, the CD molecular positive, HER2 positive, and PAM positive EV subset counting could be used for the breast cancer diagnosis. And if we change the specific protein payload, we choose the GP33 to another one payload, it could be used for the hepatoma carcinoma diagnosis as well. In a word, the DDI-PCR assay is a versatile system for the EV subset analysis at single EV level and having great potential in the cancer diagnosis. Finally, I would like to show my heartfelt thanks to the founders, the lab, and my supervisor, Professor Zheng, and my co-operators. Because the time is limited and maybe more details, we could have a further talk at the poster session. The poster number is 116. Thanks for your listening.
Thank you, Lucian. Our next speaker is Afsareen Banu. She is from Maharishi Dayanand University, Rohtak. She will be talking about proteomics of salivary exosomes, a potential breakthrough for screening of oral cancer among tobacco consumers. Welcome, Afsareen.
Thank you so much. Good morning, everyone. I am Afsareen. I am from Maharishi Dayanand University, Rohtak, and I am presenting my talk over the topic proteomics of salivary exosomes, a potential breakthrough for screening of oral cancer among the tobacco consumers. Okay, as we know that the oral cancer is one of the most common cancer worldwide, and it includes the cancers of the oral cavity and the other parts of the mouth. And as per the report of the International Agency for Research on Cancer last year, they developed a handbook on the oral cancer prevention, which was prepared by the hard work of the 25 scientists from the 14 countries worldwide. And it stated that by avoiding or stopping the tobacco smoking, it significantly reduced the chances of the risk of the oral cancer. And worldwide, we know that most of the patients with oral cancer, they are presented at the advanced stages with poor prognosis. So it is due to the lack of any early screening method, and early screening method target the high-risk population, and it will going to reduce the oral cancer incidence and mortality. And to address this cause, we are choosing the stars of the ICIP, that is the EVs, and we are choosing the EVs which have the endogenous pathway. And we know that these EVs, they have an important role in the oral cancer progression, initiation, metastasis. And to address these causes, we formulate 4 objectives, and for this we first isolate and characterize the salivary exosomes from the samples. We collect from 3 study groups, and then we estimate the total protein and expression of the cytokines, and then we evaluate the amino acid composition, and then finally we perform the FTIR spectra. And this is a brief methodology we follow, and we try our best to follow all the guidelines of the MyCell 2023. And we successfully isolate our EVs, small EVs, that is exosomes, and we characterize them through different techniques, and we found significant difference in the size of the EVs among the 3 study groups. And later, we've also found significant difference among the smoker and the non-smoker group in the concentration of the IL-6 and IL-8. And later we performed the correlation analysis with the smoking parameters, and we found that in case of the oral cancer, the parameters, they are significantly correlated with the concentration of the cytokines, but in case of the smoker group, only the IL-6, it shows a significant correlation with the smoking parameters. And as we know, FTIR is making its impact in cancer studies as shown by several papers as shown here. Okay. So we performed the FTIR analysis, and there are many regions for the protein analysis, amide 1, amide B, and amide 1, 2, 3, 4, 5. But for this study, we focus main goal to amide 1, amide 2, and amide 3. And when we compare the FTIR transmission among the non-smoker and smoker groups, we found significant results. And then we compare the relative intensity ratio of the FTIR transmittance, and we again found significant difference among the smoker and the non-smoker group. It means it has the potential to discriminate among these two groups. And then we perform the amino acid analysis, and we find a significant elevation of the amino acid concentration in smoker and oral cancer groups as compared to non-smoker group. And then we perform the FTIR relative peak intensity ratio of the amino acid side chains, and again we found a significant difference among this non-smoker and smoker group. And as this is a short talk, we are including only 3 amino acids here, and this is the unpublished data. And finally, in conclusion, we found a positive correlation between our tobacco consumption and the IL-8 concentration. And the smokers' FTIR protein spectra, it was quite similar to those of the oral cancer cases, and FTIR transmitting intensity ratio of the amino acid also distinguished the smokers from the non-smoker. And it is required to validate data with the large population set. And this is the social impact, early intervention, targeted screening of the high-risk groups, and public awareness, and ensuring timely diagnosis, and healthcare resources that can be directed towards the high-risk groups, and it will alleviate the anxiety and stress of the late-stage diagnosis. And this is the future perspective, validation of the large population set, this is references, and with this, we can't ignore the significance of these minute but significant changes, because as EVs they are small but they have a significant and they have entire array, irrespective of their size.
Thank you, Upstream. So our next speaker is Ryosuke Yukesa. He's from Nagoya University Graduate School of Medicine, and talking about analyzing coffee number variation status in extracellular vesicles as novel clinical biomarkers of high-grade serous ovarian carcinoma. Welcome, Ryosuke.
Thanks, Dr. Jorgensen and Dr. Nia. It is a great honor to be able to have a presentation today. I'd like to talk about the importance of copy number variations of EBDNA in ovarian cancer. First, ovarian cancer ranks fifth in cancer deaths among women in the USA. The main subtype of ovarian cancer is high-grade serous ovarian carcinoma. Most patients with this type of ovarian cancer are diagnosed with late-stage disease, so the mortality rate is very high. Therefore, improving outcomes in advanced cases is very crucial. Next, the presence of DNA in exosomes was first reported in 2014, reflecting the genomic information of tumor tissue. Our group previously reported that CNPs in exosomes from ascites reflected the status of ovarian cancer tissue. However, the clinical utility of EBDNA remains largely unexplored. Therefore, our objective is to reveal the potential of EBDNA as novel biomarkers for high-grade serous ovarian carcinoma. EBs were isolated according to the standard differential centrifuge method. Then, it was confirmed that EBs EVs were properly collected by nanotracking analysis and cryogenic electron microscopy. According to Nature Genetics in 2013, the genomic data of various cancer types from TCGA were analyzed, and the main genomic alteration of high-grade serous ovarian carcinoma were copy number variations and TP53 mutation. Furthermore, the ovarian cancer moonshot project revealed dominant CNV-related genes. This is the method. Comprehensive CNV status was analyzed by whole genome sequencing, while the CNV status of specific genes were analyzed by droplet digital PCR, which is characterized by high precision and sensitivity. 30 genes were selected as target gene panel for ddPCR analysis based on the ovarian cancer moonshot database. This is the result of whole genome sequencing. CNV status of EBDNA in ascites reflected the status of tumor DNA. However, the status of EBDNA in serum didn't reflect that of tumor DNA, probably because the concentration of DNA derived from the tumor was very low. This is the result of CNV analysis for cell lines using ddPCR. 2 cell lines derived from high-grade serous ovarian carcinoma were used for evaluation, and both cell lines showed a high correlation of CNV status between EBDNA and cellular DNA. Next, we examined clinical samples. EVs were collected from 1 milliliter of serum or ascites. Similar to the result of whole genome sequencing, CNV of EBDNA in ascites reflected the status of tumor DNA. Here we examined the correlation according to the size of EVs for 3 patients with high-grade serous ovarian carcinoma. The status in small EVs in ascites showed a higher correlation with the status of tumor DNA in all cases. This is probably due to differences in EV synthesis. So far, it is shown that we can get information on CNV on tumor DNA by analyzing EBDNA in ascites. Next, we consider the clinical applications of this assay. First, the copy numbers of normal ovarian tissue and ovarian cancer tissue were measured and compared. As a result, normal ovarian tissue had inherent copy numbers, and the copy numbers of the genes shown in the slide were significantly higher in malignant tumors. Next, CNV evaluation was performed by ddPCR using 1 milliliter of undiluted ascites from patients with benign ovarian tumors and ovarian cancer at the time of surgery. As shown in the figure, very little DNA was present in ascites from benign patients, suggesting that increased DNA in ascites itself indicated the presence of malignancy. We next examined whether the amount of DNA in ascites differed according to the result of ascites cytology. The concentration of RPP30, a housekeeping gene, was measured. There were cases whose amount of DNA was elevated in ascites with cytology negative, as well as the cases cytology positive, suggesting that the amount of DNA in ascites increases from earlier stages. Then, there arises a question that where the DNA released from the tumor cells exists. Cell-free DNA is present as EBDNA or macromolecular complexes in body fluids. In general, cell-free DNA is considered to be floating DNA and easier to collect and analyze, while EBDNA is thought to be more stable and contain longer DNA. We previously reported on the mechanism of DNA loading into exosomes. There were significantly more DNA-positive exosomes derived from malignant cells. Similar results were observed in micronuclei. Electron microscopy images show that micronuclei were released from the nucleus and then collapsed and merged into early endosomes. In this way, we have shown that DNA may be specifically loaded into exosomes via micronuclei. This is consistent with the result of decreased DNA in ascites of ovarian cancer patients. Therefore, we assume that cell-free DNA is mainly present in EVs if cancer cells specifically release DNA into EVs. As shown in the slide, we define the conventional cell-free DNA as total cfDNA, DNA obtained from the ultracentrifuge pellet as EBDNA, and pure cell-free DNA obtained from the supernatant as cfDNA, and then measured these CNVs by ddPCR. This is the result. The CNV status of total cfDNA and EBDNA showed high correlation with the status of tumor DNA, while cfDNA showed a low correlation. Therefore, cell-free DNA derived from tumor cells may be mostly in EVs. Lastly, we investigated whether CNV status could predict treatment response. PARP inhibitors have become important therapeutic options for maintenance therapy. In general, BRCA status and homologous recombination deficiency are considered predictive biomarkers of response. However, it is challenging to identify responders in the real world, so there is a strong demand to find new biomarkers. Here, we investigated the relationship between CNV status and response to PARP inhibitor treatment. Patients with high-grade serous ovarian carcinoma who were treated with olaparib were included in this study. Responders were defined as patients who received olaparib continuously for more than 1 year, while non-responders were those who discontinued the drug within 6 months due to recurrence or disease progression. Patient characteristics are shown in the table. The responders included BRCA-negative patients, and both groups included chemotherapy-naive and post-chemotherapy samples. Then DNA was extracted from FFPE tissue at the time of surgery. Then CNV analysis was performed. As shown in the heat map and PCR analysis, CNV profiles were different between the groups. The predicted performance of each gene was evaluated by cross-validation, and the top 9 genes were selected as candidates for predictive response. The profiles of the heat map were clearly different, and responders tended to show higher copy numbers. The ROC curves for each gene showed good AUC values. Furthermore, a combination of 4 genes resulted in excellent prediction whose AUC was 0.98, and adding 2 more genes resulted in an AUC value of 1.0. Thus, CNV analysis could serve as an alternative predictive biomarker for response to PARP inhibitors. To validate the potential of the selected genes, CNVs in EBDNA from 2 cell lines were measured. The results showed that higher sensitivity to olaparib was associated with higher copy numbers. In clinical samples, EBDNA in ascites at the time of surgery was collected from 4 patients who received maintenance therapy with olaparib. 2 cases were continued for more than 1 year, while the other 2 cases were discontinued for less than 1 year due to disease progression. The copy numbers tend to be higher in the cases with longer duration of the treatment. In addition, the prediction equation based on the 4 genes correctly predicted the treatment response. In particular, it was able to identify the treatment resistance of patient 3 whose HRD was positive. In this way, the selected genes were validated using EBDNA both in vitro and clinical samples. This is a summary of this presentation. We demonstrate that tumor CNV status could be detected in EBDNA using ddPCR method. We also demonstrate the potential of CNV analysis as diagnostic and prognostic biomarkers. We would like to thank all involved in our study. Thanks for your attention.
Thank you. Do you have any questions from the audience?
Thank you so much for an excellent talk, excellent. I'm wondering, so micronucleus could be required. related to the exosome-related DNA and showing the copy numbers. Is it also related to the kind of some autophagy, like some process to like some degradate such a micronucleus? Because one of the aminopathy, it's a mutation in the nucleic membrane showing like some autophagy directly towards the injured nucleic membranes. And if like some autophagy, like some activities decrease, then related to the release of the micronucleus into the bloodstream would be kind of suggesting kind of some tumor, like some metabolic like some conditions, and potentially you can find and stratify the patient based on such like some other pathways, activities, or drug target. And, so, do you have any ideas?
Thank you for it. Very question. Indeed, we have tested the mechanism, the micronuclear change. So, but it may be related to the mechanism.
So, additional question related to that. So, is there any like some difference in terms of the number of the double-stranded DNA in the EVs compared or with the tumor size or tumor grade, something? So, small tumor size, however, like so much amount of the double-stranded DNA positive EV may suggest something in the tumor future.
Thank you. We check the amount of DNA according to the size. There are almost same volume of DNA in small tumor or large tumor. So in ascites, we can detect DNA so we can diagnose ovarian cancer.
Thank you so much.
I have a quick question actually. What do you mean by, like, you said that CFDNA, cell-free DNA is present in EVs?
Thank you for the question. Maybe cfDNA is the result of apoptosis cell, so less stable and contain shorter DNA. So EBDNA contain, if EB specifically released from tumor cell, we can get more informative EVs.
Exactly. You're like saying the difference, yeah? So I'm sorry to say that the CFDNA and EVDNA, they are like completely different. So maybe the reason why you find CVDNA in your EV preparation is because of ultracentrifugation, where you get EVDNA as well as CFDNA in your preparation. I mean, I'm coming from University Hospital in Germany, and so we have a paper which got published like 2 years before, and we use size exclusion chromatography, and in that, like, you get EVDNA like in different fractions and cell-free DNA in different fractions. So maybe you can refer that. This will tell you. You were telling the differences exactly. So EVDNA is like more specific cancer, like where you can get cancer-specific information, from the EVDNA, but not from cell-free DNA. I just wanted to correct that. Okay. Thank you.
Thank you.
Thank you, Bruce. I would like to let's give an applause to Rose for a great talk, and I'd like to invite our next speaker, Thomas Steckmeyer from Monash University, talking about extracellular vesicles as biomarkers of endometriosis, a comparison between peritoneal fluid and peripheral blood. Welcome, Thomas.
Thank you. Hello, everyone. I'm Thomas Steckmeyer from Monash University right here, and I think the organizers will give me a chance to present on EVs as biomarkers of endometriosis. Can I have a quick show of hands first? Who does not know what endometriosis is? All the men raising their hands now. It's a disease in need of a biomarker, I can tell you. So let's click this forward. Briefly on endometriosis. Endometriosis is a condition characterized by the growth of endometrium outside of the uterus. You might know that it builds up and bleeds off monthly in the menstrual cycle, and the tissue that goes outside of the uterus and the peritoneal cavity does the same thing. And because the blood cannot escape, it causes chronic and acute inflammation, severe pain during menstruation and in between, and has a high rate of infertility accompanying it. There are no really good treatments at the moment. You can try to take painkillers, you can shut down the cycle altogether, or you can try surgery and remove the tissue that has grown elsewhere. But because surgery is not the first thing you do, it's very difficult. People have to wait a long time before they get diagnosed correctly and maybe have the tissue removed. And even then there's only 50% success rate in ameliorating the symptoms. Endometriosis affects a lot of people, 10% of women of reproductive age worldwide, so it's really prevalent and causes a considerable healthcare burden in Australia and globally. Important for us to notice is there are 3 different kinds of endometriosis. There are ovarian cysts growing on the ovaries. There's deep endometriosis, that's nodules formed deep within the tissue, and superficial disease in the peritoneal cavity called dot clot disease. If you know flow cytometer, you know why it's called that. And they are all different in terms of the diagnostic need. So there is a long diagnostic delay in endometriosis, as I mentioned, because of this need for laparoscopy to make a decisive diagnosis. However, recently the guidelines of the European Society for Human Reproduction and Embryology, ESHRE, have been revised saying you can actually diagnose ovarian cysts and deep endometriosis using ultrasound and transvaginal imaging. But you cannot see the peritoneal lesions using those methods, right? Because they're too small, and we need a biomarker for those lesions. People have looked at all kinds of things, cytokines, cells, mRNA proteins, and so on, and we thought we look for a biomarker that is ideally produced by the lesions where they are, i.e. in the peritoneal fluid itself, because there you would expect the highest signal-to-noise ratio of anything you look at, right? That's right where it comes from. So in the most proximal tissues or fluids concerned, this would be the peritoneal fluid, and whatever we find there during the discovery stage, we then aim to translate into peripheral blood testing to see if you can trace the same biomarkers in blood, because if you have peritoneal fluid as a sample, that's useless because you have to do an operation anyway to get it. So you might as well then diagnose the person. And in the end, we have a validation stage to test animal biomarkers. I don't think I need to explain to anyone here what these are and why they're useful, so let's just move on from this. We'll be looking at protein cargo, and the first thing we did was do the gradient with different fractions, and you can see here, so sequential centrifugation steps to remove cells, microvesicles, and then pellet vesicles in the gradient, watch them analyze fractions, and this is the refractory index versus weight to determine the density of each fraction, and then you can have a high density and low density peak in your particles, and here are TEM images of that. Here's the density gradient. There was a paper in 2015, I believe, by Edward Lewis et al. Describing this method, so he followed his example. This was still at Oxford, I moved to Monash later, but we'll come to that. One of the problems we found is that when you use these gradients for most other methods that you've tried, we end up with very pure amounts of nothing, so there's almost never enough to do any downstream analysis like proteomics or looking for different biomarkers. And so we tried idexinol size exclusion chromatography. We filtered off the larger vesicles and just kept the smaller ones. The only thing that seemed to be good at delivering enough material was ultracentrifugation, I'm afraid to say. I also tried Exomet. I don't know if Exomet Innovic is here at the moment. They keep trying to persuade me to try again. We'll see. The disadvantage is that we cannot do NTA on the positive fraction, right? So we don't really know what we have. So peritoneal fluid as a proximal sample fluid is good for discovery, to see what the biomarkers might be you look for, but it's very impractical then for screening or any testing you have to do. And of course the vesicles we find in there can also come from anything accompanying the lesions that form, like inflammation and so on, or endometrial cells that bleed retrograde into the cavity. And our ultimate goal would be to biomarkers. So So this project started some time ago at Oxford as the master's and then DPhil project of Hannah Nasri, who had a poster yesterday. So the plug is a bit late now, but I hope you went and talked to her. She did a pilot experiment to show that we can actually analyze, isolate EVs from peritoneal fluid directly without culturing anything in between, and then do proteomics on the contents. And she has done since the discovery project that for the first one, she had pooled samples, had enough material. Then we did a discovery study where she analyzed individuals and the EVs therein for the protein content. And at Monash, I have since had 4 very talented honor students driving the project forward: Johanna Farley, Natasha Borash, Chloe James, and Claire Davison. And there we collect samples, blood samples and peritoneal fluid samples from the same patients to then try and trace biomarkers. And we have so far built up a bank of 106 paired sets. Sorry, is this about right? Yeah. In the first study, Hannah was able to show that yes, we find these in different cycle phases. So remember, we have to throw the menstrual cycle into all of this. So we distinguish the menstrual phase, proliferative phase, and secondary phase of the endometrium, which has implications for the lesions, how they build up and bleed off. And then we distinguish between endometriosis, mild and severe stages, and control patients. So we did electron microscopy, NTA imaging for the tetrasparmins, Western blotting for Susan and Alex, and then proteomics. Just briefly, if you look at this graph here, so these are the different patients that went in, NTA data here, and the amount of vesicous changes with the menstrual cycle. We didn't have any menstrual women in this study yet. We have them now. So you have more vesicous generally in the proliferative phase, when the endometrium goes up in the secretory phase. And interestingly, there are a lot more in what we call stage 1 or 2 mild disease than in stage 3 or 4, which is severe endometriosis. And the reason is probably that the staging system doesn't have a biological logic to it. It's just the surgeon determines the stage by what he sees in laparoscopy and taking into account the other symptoms of the patient. So it doesn't necessarily develop from 1 to 2 to 3 to 4 as you would expect in cancer. So stage 3 and 4 are characterized by the endometrioma and the deep nodules, whereas the superficial disease, the dot plot disease, is usually stage 1 to 2. And I think therefore it makes sense that the lesions that are present in the peritoneal cavity might just give off a lot more vesicles into the liquid than the deep nodules can, because they are deep in the tissue. The vesicles never reach the peritoneal fluid. Right? We then did proteomics on the next phase. This was the individual study. There she had 120 controls in 45 cases, and then different groups again. And you can nicely separate them, the controls from the endometriosis cases, by proteomics. And the results were on posters, so she can then identify different groups of proteins that are endometriosis specific versus controls. At the same time, at Monash here in Australia, we have been collecting blood samples and peritoneal fluid samples, and then also did proteomics on it, and the PCA here shows a nice separation between the different tissue types. Let's look at menstrual fluid with Shanti Puram, who might be sitting in here somewhere. He's too shy to wave at the moment. And then you get these volcano plots of proteins up and down regulated between cases and controls in peritoneal fluid and in blood. Then we looked for biomarkers in these proteins that we were given from the proteomics people. And the ideal biomarker, we thought, is something that's normally not expressed in peritoneal fluid or in blood, ideally. And when in doubt, we would look at peritoneal fluid expression over the proteomics of blood, because you have much more, you have less sensitivity in proteomics than you might have when you trace these with an antibody. You don't have the same depth in reading protein data. And that means, of course, there have to be antibodies available for these markers. And we have 2 candidates, and I'm sorry, the business development people tell me I can't tell you what they are, so maybe for now. And Natasha Borash, an honor student, did a nice Western blot on these. And the next student, Chloe James, then tried to use automated Western blot go back to traditional Western blotting, and you can tell that's not our favorite method. But we looked at sentinel positive samples and then whether or not the candidate is present in traces or not. And just because Marlene had mentioned the AUC curves and true and false positives and these kind of calculations, we went ahead and did these calculations anyway based on the Western blotting data, and you get a certain sensitivity and specificity. And the challenge is now to translate that into the capillary Western blotting system, protein simple, because that uses a lot less protein for A1. There's only 10 micrograms for normal Western blot. You can get away with 0.5 or something, a lot less. And if you remember the cutoff we had at the beginning between the different methods, if we could use this system, it would mean we can probably afford to look at more pure preparations in the future. So we expected our candidate B to show up here in this lane, which it didn't, and we had a lot of mystery bands here. Not sure what it is, probably albumin or some other common blood serum protein, plasma protein, sorry. We are not that interested in what it is, we just want to get rid of it. So we then tried to do the SEC-isin separation after the ultracentrifugation, so different fractions here. You have vesicle numbers and protein amount. And here again, the idea that you have a few big proteins that are very common and you have to get rid of them. I think you're all familiar with the difficulties of this sort of thing. And I've since for syntonin in the fraction 3, the vesicle fraction, and fraction 10 is the protein fraction, and you can see that syntonin shows up nicely in these dilutions. So we're currently trying to find out how low we can get with the dilution to still get a signal for syntonin, and then we'll try the markers again. We are aiming for 10 samples in each group, so menstrual, proliferative, and secretory phase of the menstrual cycle, and then control for endometriosis, ideally mild endometriosis stage 1 or 2. And we're almost there, so we need a couple more samples here, and then we can do the overall analysis and just test everything. I'd like to finish with a caution perhaps. So this is a comment we got back from reviewers, not from the EV field, because we are all crazy about accessory vesicles, but from the clinicians in Oxengani who said, this is just the next hype, perhaps after we've heard prostaglandins are the thing for diagnostics, or cytokines, or then stem cells, genetics, systems biology. So, I do hope we're proven wrong with data eventually, but I think it's important that you remain your own best critic in this matter. Thank you for your attention, and I'd like to thank my sponsors from all over the world, actually, and the people who've made this project possible at Oxford and Monash University. Thank you. Questions?
Thank you so much for a great talk. I think we have just time for one single question. Anyone?
Can I just ask? Yeah, great talk. I did once upon a time worked on endometriosis when I was in the University of Maastricht in Netherlands. So it's nice to see the field is evolving. I just had a question. So you know that women and women with syndrome, these are women at risk of developing endometriosis. So when you did the control and the endometriosis group, when you are comparing the proliferative and the secretory, did you also compare like BMI and try to match these two cohorts?
We haven't correlated that yet, or we do exclude people with a severe BMI one way or another. And previously, when looking at the UK Biobank data, we found that women with low BMI are more prone to get endometriosis. So it seems to be different in different populations, as far as I can tell. But it's an interesting idea.